In this lab, you will use the microscope to observe eukaryotic and prokaryotic cells, as well as several unicellular and multicellular organisms. Before you can do that, though, you need to become familiar with how to set up a microscope, the main components of the microscope and how to use them, as well as how to determine the size of the field that you're viewing so that you can actually estimate the sizes of the cells or the organisms that you're observing under the microscope. In this video, you will see Emily setting up the microscope, and I will pause the video as I go through it so that I can kind of explain things as we go along. So first thing that Emily does is she picks up the microscope, and you'll notice she does it with two hands, which is very important because the microscope is quite heavy and you don't want to drop it and hurt yourself or break the microscope. She's now removing the cover and then plugging the microscope in. You'll notice that the power cord is actually tucked behind the microscope, so make sure you wrap it up and tuck it behind the microscope again for storage. The on-off switch is on the right-hand side of the microscope, and the same switch also controls the intensity of the light. Make sure to put the light on the lowest intensity that allows you to still view the image, but it's not so bright that it's hurting your eyes or hurting the specimen, especially if you're working with a live specimen. So the lower the intensity, the better. You also see that right now Emily is holding the eyepiece in her hands. So that's the piece you would actually look through, and that adds on 10 times magnification to your total magnification. So make sure you don't forget to include the eyepiece magnification when you're calculating the total magnification of the microscope. And the eyepiece can actually be adjusted for the width of your eyes. That's important. Now Emily is flipping through the objectives. We have 4 on these microscopes, 4x, 10x, 40x, and 100x, and you always start on the lowest. Emily is now also turning the knob to adjust the stage height, which is important for when you're positioning the slide and bringing it into view. So again, always start on the 4x objective. So now Emily is taking the micrometer slide just to demonstrate how a slide is positioned. This will be done the same way for any slide that you're using in the lab. And you'll see there's a little lever on the left-hand side that kind of pulls out and allows the slide to be tucked into place. So make sure you don't force that and don't put the slide under the lever, which is a common mistake that students make. It has to fit right next to the lever, and the lever kind of just holds it in place. You'll also notice that Emily is now adjusting this knob underneath the stage on the right-hand side. You'll see it when I start playing the video again, to bring the specimen that you want to see right into the center of the view. So it should be positioned right underneath the objective so that you're more likely to find it when you're looking through the eyepiece. And then, of course, you can, once it's in the center, you can then start bringing the stage up and down as you're looking through the eyepiece to actually get your image into focus. Okay, so Emily's positioning it as in the center, and that's how you would actually look for the specimen. The other device you'd be using in this lab is this little ruler. The ruler is not a proper slide, so that one just kind of sits on the stage without being, you can tuck it under the lever, but it's not really held in position properly like a normal microscope slide would be. You just need to place it so that you can see the little tick marks underneath as you look through the eyepiece. Okay, so now that you've set up the microscope, the first part of the lab is to figure out the sizes of the fields that you're observing under the different objectives. For the lower power objective, for the 4x, you're using the ruler, as we saw in the video, the last thing that Emily put on the microscope. So the ruler is shown on the left side here. And then the view that you would see when you look through the eyepiece with the 4x objective is shown on the right. So to determine the size of the field, you would just count how many spaces between the tick marks you have, and then essentially just determine how the distance or the diameter of your field of view. As you go higher in magnification, if you continue looking at the ruler, first of all, your image will be very fuzzy, but also you will start not being able to tell how many spaces you actually have, just because the ruler, the tick marks are positioned too far apart for the higher objectives. So what we use instead is this micrometer slide, which you saw Emily use in the beginning of the microscope demo. And you can see it very well under the 4x objective on the left. So it's tiny compared to the ruler, as you can see. And that 4x objective is actually not very useful because it doesn't even span the entire field. However, when you use it at 10x or 40x objective, you can clearly see and count how many spaces between your tick marks you have, and this way you can estimate the diameter of your field of view. Once you know the diameter of the field of view, then it's very easy to determine the approximate size of your cell or specimen that you're looking at by kind of imagining how many of those cells or specimens would fit into your field of view, and then taking that number, taking your diameter of the field of view, dividing by that number, and kind of getting the estimate. So you will use these images that I showed on the previous slide and this slide if you're doing this lab remotely to actually answer the questions in the first 3 parts of the lab. But of course, if you're doing this lab in person, you will get to see your own images and can answer the questions that way. Okay, so I'd like to again thank Emily and Aaron for helping set up these demo labs, and I hope you found this information helpful. Thank you.